Today we're going to begin talking about protein synthesis, and this is actually going to be a two-part video. The goal of this first part is to explain how genes in DNA can be transcribed into mRNA in a process called transcription. Okay, so first let's talk about genes and how they code for proteins. A gene is a section of DNA that codes for a protein. And the process in which cells use genes to create proteins is called protein synthesis. The cell is going to use those proteins for a variety of purposes. One is to transport things around the cell, another is to give the cell structure, and another is to speed up chemical reactions. Proteins that speed up chemical reactions within the cell are called catalysts. So these are only some of the things that the proteins are going to do inside the cell. All right, so how exactly are proteins made? This is the big picture. There are basically two steps of protein synthesis. In step one, you go from DNA to RNA, and that step is called transcription. In step two, you go from RNA to proteins, and that step is called translation. So in the remainder of this video, I'm only going to be talking about what's happening during step one, which is transcription. In the next video, we'll talk about what happens in step two, translation. Okay, so what happens during transcription? Well, in eukaryotes, so these are cells that have a nucleus in them. In eukaryotes, transcription occurs inside the nucleus, where the DNA is at. And there are going to be three basic steps of transcription. There's initiation, elongation, and termination. So this picture you can see labeled initiation, elongation, and termination. So in this one picture, it's kind of showing you from top to bottom the three basic steps that's happening in transcription. And I'm going to go through some of the specific events that are happening in each step now. Okay, so what happens during initiation? Well, during initiation, the enzyme called RNA polymerase binds to a region of the gene that's called a promoter, and then the DNA unwinds. So in this picture, you can see RNA polymerase is this purple blob here. It's going to bind to a specific region of the DNA called the promoter, and then the DNA is going to unwind. So that's what happens during initiation. Okay, now let's move on to elongation. So during elongation, that RNA polymerase is going to start adding nucleotides, and by doing that, it's going to build a complementary strand of RNA. So this purple strand that you see here has these blue tick marks attached. Each tick mark represents a nucleotide that's being added, and that strand that's being built is RNA. So during elongation, we're building a strand of RNA. All right. There are some base pairing rules that you need to be familiar with during this step. So as nucleotides are added, they should complement the strand of DNA. So here are the base pairing rules. Say that I have a strand of DNA here with the letters A, G, C, T on it. When that strand of RNA is being built, U in the RNA is going to pair with A. C in the RNA is going to pair with G. G with C, and A with T. So notice as we're building the RNA, all the base pairing rules that we discussed in DNA replication still apply, except we don't use the letter T in RNA. So if you ever catch yourself writing the letter T while you're building RNA, you've made a mistake. It should be a U instead. Remember, RNA uses a U instead of a T. So those are your base pairing rules when you're building a complementary strand of RNA. All right, now let's go to this last step of transcription. This step is called termination. So during termination, RNA polymerase crosses a stop sequence in the gene, and that's what you see in green here. That represents the stop sequence in the gene. And when RNA polymerase crosses that stop sequence, the RNA polymerase and the strand that's being built detach from the DNA. And so that's why you see the purple blob and this purple strand detaching, moving away from the DNA. Okay, so we just discussed initiation, elongation, and termination. Now, that's not quite the end of this process. So after you've produced this complementary strand of RNA, it has to be processed. So before that RNA that you just built, that strand of RNA that we just built, before it can leave the nucleus, it has to be edited. Introns are going to be spliced out. So I use the word spliced out to give you an idea of like cutting it out and removing it. So introns have to be spliced out, and the exons that are left behind are going to be joined together. So in this picture, you can see green sections of the RNA and yellow sections of the RNA. The yellow sections are the introns, and notice they're being removed. And the green sections represent the exons. The green sections are left behind, and they get joined together. So my mature form of RNA will have exons in it, but no introns. All the introns will be removed. Now you may think, wait, what's an intron? What's an exon? Well, introns are just regions that do not code for protein. So these yellow portions, they represent regions that are not going to be used to code for a protein. Exons are regions that do code for a protein. So what we're left with are all the regions that code for protein in our mature form. The regions that didn't, we've removed. Okay, there's one last step to do to the RNA, and that is polyadenylation. So there's a tail of the letter A's, which are adenine bases, that get added to the end of the RNA. And that process of adding that string of A's at the end is called polyadenylation. So you can kind of see in this picture what we have. So this would be my strand of RNA. And at the very end, we're going to add a whole lot, more than just 3. There's going to be a whole lot of A's, the letter A's here at the end. And that string of A's signals the end of the mRNA. And the string of A's are actually involved in exporting the mRNA from the nucleus. Now, I've used the word mRNA here, so you might wonder what that M stands for. So at the end of this process, at the end of transcription, we have a piece of mRNA. And that M stands for messenger RNA. And that mRNA is a copy of the set of instructions that was stored in the DNA. It's a copy of those set of instructions on how to build a protein. And the mRNA is going to leave the nucleus and carry this message, the message being these instructions on how to build a protein. The mRNA is going to leave the nucleus and carry the message to the ribosome, where translation will take place. So in this picture kind of summarizes what we've seen so far in this video. We start with a piece of DNA, and we're going to build a piece of RNA with it. Notice that RNA has sections that are that code for proteins. That's going to be the red sections here, the exons. And it also has green sections. Those are going to be the sections that do not code for a protein. Those are the introns. So once you do transcription, you've got this piece of RNA with exons and introns in it. You're going to splice out the introns and join together the exons. Then you're going to add a string of A's to the end, and that represents the end of transcription. So in our next video, we're going to see how this mRNA that we built, this messenger RNA, is going to leave the nucleus and go to the ribosome where translation will take place.